Okay, this video is to help you with your Dragon Genetics Independent Assortment and Genetic Linkage assignment. On page 1, you are to just show the phenotype on the mother dragon and the father dragon. And as you can see on my page, my phenotype is drawn in, and I use colored pencils to draw in the wings and the horns, and I colored them in. And you can do that too, and you don't have to copy the way I did it. You can be creative and draw your own wings and your own horns like you want. So that's the main thing on page 1. On page 2, you're looking at how the chromosomes are separated during meiosis. And the example on the page has you draw your own chromosome separation out for meiosis. So make sure you have one cell starting, and at the end you have 4 independent cells. Now when you get to page 3, at the top, there's a table. And again, on this table, you're looking at how the traits were separated when making the different dragons. And so in the picture, you're to move the chromosomes and give the genotype and then draw in the phenotype, which I've done again with wings and horns by using color pencils. In the table below, what we have here is now we have what's called a dihybrid cross. A couple things that I did on my dihybrid cross to make things easier for me to see is I highlighted a row and I shaded in 2 columns. Because the father is homozygous for both traits, the rows will all be the same, the columns will all be the same. It's the mother's who's heterozygous, so we don't have to play around with a whole lot of things. So I use that to help me answer the questions that go with it. On page 4, you're going to do an activity, and it tells you to take 2 popsicle sticks. Well, you don't need 2 popsicle sticks. You can use a penny and a dime. And what these represent are the chromosomes. Okay? The traits, the alleles for the trait on the chromosome are on the different sides of the penny. I have heads representing the dominant trait and tails representing the recessive trait. Okay, I'm going to have wings on my dime and I'm going to have horns on my penny. So in order to simulate independent assortment and random selection, I'm going to hold my penny and my dime about 6 inches up, and I'm going to hold them and I'm going to drop them both at the same time. Okay, what I inherited from my first dragon baby is what I see when my penny and my dime land. Here I have 2 heads. That means I have 2 dominant alleles. So my first child is big W, big H. In the table, I'm going to put a tick mark, just like we did with the penny genetics. I'm going to put a tick mark for big W, big H. I don't have to worry about the father because he's giving me the little w, little h. So this just represents the mother dragon since she is heterozygous. I'm going to repeat this process 3 more times, adding tick marks. Falling off the table, so I'm going to do it again. Each time I do it, I create another baby. So this baby has heads on the penny, which remember represents horns, and tails on the dime, which represents wings. So I'm little w, big H. So when I combine with the father, I'm little w, big H, little h. And I'm going to put a tick mark in my table for that genotype. Now I'm going to complete this 4 times. Then I'm going to go to page 5. If I can find my page 5. On page 5, it wants you to compile the data for all the baby dragons produced by all the students. However, we're not doing this in class, so you cannot compile the data from all the students. Just do your 4 baby dragons. Okay? What it wants you to do is first give the genotype of the baby. For the genotype of each baby, you're going to have a 4-letter genotype. You're going to have 2 W's and 2 H's. Keep your W's together and your H's together. Remember, always put a capital letter first and the lowercase letter second. Then you're going to put a check mark for the phenotypes that baby will display. For example, wings or no wings. bighorn or smallhorn. Then you're going to answer the questions that follow. On page 6, you're going to fill in your own dihybrid cross at the bottom of the page. I filled in mine. When doing a dihybrid cross, you want to keep the same letters together. For example, I want to keep my S's together and my X's and Y's together. So I would have big S, big S, big X, big Z, big X. I would have big S, little s, big X, big X. I would have big S, big S, big X, big Y. I would have big S, little s, X, Y. So that's how you're going to fill those in on your dihybrid cross. What I did with mine, as you can see, is I divided it into halves, because half of the offspring will be male and half of the offspring will be female, and it's going to help me evaluate that Punnett square a little easier. Then you're going to go on to page 7. And on page 7, you're going to look at traits that are carried on the same chromosome and then a trait carried on a different chromosome. So in the middle of page 7, you have a representation of the two different chromosomes, and you're just going to fill out the alleles. The first chromosome has been done for you. You are just going to do the second chromosome. The second chromosome should have only one letter in it. Then you are going to show the phenotypes of the heterozygous father and the homozygous mother. Again, I did mine. I colored it in, drew and colored, and make it kind of fancy. Okay? Anytime we improve things, it's always better. On page 8, you're going to go through meiosis again by filling in how the alleles are moved during the process of meiosis. At the bottom on number 3 on page 8, you're going to make your own dihybrid cross. Now, for this dihybrid cross, okay, you can see that I have 4 columns, but I only have 1 row set up. This is because the father is homozygous. This is the father or the mother. The mother, she's homozygous for all traits. So there's only one possible allele combination for her. However, since the father is heterozygous for all traits, I have 4 possible combinations. Where do I get those letters? Up here in this table, what I got at the bottom. Okay? So now when I fill this out, I'm going to carry all the letters into each box. Looks like a lot. In each box, here's how I line up my letter combinations. I keep the W's together, then I go down, and underneath those, I write the next letter combination. Then I go down, underneath those, write the next letter combination. And then the last, because we're looking at 4 different traits. The first 3 traits are carried on one chromosome, while the last trait is carried on a second chromosome. So you can fill in each box this way, so it's not so hard to read. So you have each trait listed separately. On the last page, you are going to indicate the 4 possible genotypes of the baby dragons from the dihybrid cross you just completed. You're also going to show the phenotypes. Again, I got a little creative with mine, and I put drawings and colorings in there. Remember, genotypes are letter combinations, while phenotypes are what you see. So for number 4 on page 9, the letter combinations for the genotypes, since I have 4 traits, there will be 8 letters. Okay? The phenotype will only be expressed as 1 because this is a dominant or recessive trait. Okay. On page 10, there are just a few questions for you to answer. Read through the lab very carefully, and it'll help you answer all the questions. If you have any other questions, you can text me. This is Miss Mahaffey signing off.